Hi guys, today we are talking about translation. This is the last piece that gets us from DNAs to proteins. So translation is a process by which mRNA is decoded and a protein is made. A codon is a series of 3 nucleotides that codes for one amino acid. Stop codons are codons that signal to the ribosome to end the translation process, thus ending the protein. A start codon is a codon that signals to ribosomes to begin translation. It codes for the first amino acid in every protein. And an anticodon is a set of 3 nucleotides in a transfer RNA molecule that binds to a complementary messenger RNA during translation. Translation is a process that converts or translates the DNA message into a polypeptide. But remember, that message is in the form of RNA. One or more polypeptides put together make up a protein. It's coded in series of 3 nucleotides consisting of A, T, C, or G in DNA, and A, U, C, and G in RNA. Proteins are read based off of the amino acids. There are 20 total amino acids, and you can consume 9 of them. Or you can make 9 of them in your body, which means you have to consume the other 11. In genetics, all codons or words are a series of 3 letters. So in genetics, we look at our words as only being 3-letter words. A codon is a 3-nucleotide sequence that codes for an amino acid. So right here on your test, I will give you a codon chart. Your codon chart will look something like this. You read this codon chart from the inside out. So the biggest letter is letter 1. Second biggest letter is letter 2. Then these tiny letters are the third letter. Those tend to be the least important in the codon, and your amino acids are on the outside. So in this example, right here, we have G, C, U. So we would go G, C, and then find the U, and then we find the amino acid on the outside. And that's what you would write down for your protein. This is another way of looking at it. This is a box-shaped triplet code. Some people tend to like this because it gives them the abbreviations of the amino acids. You have your first letter, second letter, and then third letter. The amino acid leucine is represented by 6 different codons: CUU, CUC, CUA, CUG, UUA, and UUG. In most cases, codons that represent the same amino acid have the same first 2 letters. The 4 codons that code for alanine, which is another amino acid, all begin with G and C. They tend to be the most important in the reading frame or in the codon because that third letter is considered something called a wobble. There are some very important codons that you need to know. There are 3 different stop codons. They signal to the end of the amino acid chain. UAA, UAG, and UGA are your stop codons. You also have a start codon, which signals to the start of translation and codes for the amino acid methionine. In translation, AUG or methionine will always be the start of your protein, but usually gets removed in a processing step. mRNA translation, codons must be read in the correct order. This is called our reading frame. Codons are read without spaces as a series of 3 non-overlapping nucleotides. One thing I recommend is every 3 nucleotides, put a line, an imaginary line or a real line right on your paper. That helps you remember that you're not overlapping any of your amino acids, because if you change your reading frame, you change the resulting protein. Proteins may stop prematurely if they read their reading frame incorrectly and reach a stop codon early in the mRNA sequence. This is why punctuation, capitalizing your first protein with methionine, and ending with a period with your stop codon plays an important role in the genetic code. Changing just one single nucleotide can change the whole reading sequence, making a different protein or making no protein at all. The genetic code is also said to be shared by all organisms. That means that UUU, which codes for phenylalanine, is a codon that occurs both in amino acids, in cacti, and in humans. There are very few numbers of exceptions to this rule, which is why the genetic code is said to be universal for this reason. It suggests that all organisms at one point in time arose from a common ancestor. Amino acids link together to become proteins. Proteins are made in the ribosomes, but how do we know this? Well, we found that mRNA is a very short-lived molecule that carries instructions from DNA in the nucleus out into the cytoplasm, that messenger RNA is read as a set of 3 nucleotides or codons, and through the use of transfer RNA to help ribosomes read the message and create the appropriate protein. Ribosomes are made of a combination of ribosomal RNA and proteins, and they catalyze the reaction that forms bonds between the amino acids. They have a large and a small subunit that fit together and pull the messenger RNA strand. The small subunit holds the messenger RNA in place, and the large subunit holds onto the growing protein. Ribosomes inside have something called transfer RNA. Transfer RNA acts as an adapter or a magnet between the amino acid and the corresponding messenger RNA codon. It carries free-floating amino acids from the cytoplasm and into the ribosome. They're typically folded into an L shape where one end connects to the amino acid and the other connects to an anticodon that acts as a magnet to identify its correct codon. Translation has many steps, and it takes a lot of energy from the cell. It happens in the cytoplasm when the small ribosomal subunit binds to the messenger RNA in the cytoplasm. This signals a large subunit carrying a methionine attached to it binds to the start site to find the AUG start codon. This binding seals in the larger subunit. The ribosome then pulls the messenger RNA through one codon at a time, adding the appropriate amino acid with each. It shifts through the ribosome until the messenger RNA has been fully read and the protein is produced. These are just the steps broken down. So first, the exposed codon attracts the complementary transfer RNA molecule bearing the amino acid. Ribosomes help form the peptide bond between the 2 amino acids, and then break the bond between the transfer RNA and the second site and its amino acid. The ribosome pulls the messenger RNA the length of one codon. Transfer RNA sits in the second site, then shifts to the third site, which is called the exit site. Transfer RNA leaves the ribosome to return to the cytoplasm to pick up a new amino acid, and the process continues until they reach the stop codon.